All right, well, hello everyone. I'm here today to perform the cell membrane model lab for you. And so let me just kind of go over the supplies real quick and talk a little bit about safety and things like that. But really we just have some water here, and I've got just a cup to represent our environment that we're going to put the cell in. We've got some iodine. Remember, iodine reacts with polysaccharides and turns sort of a dark purple or a black color when a polysaccharide is present. Our baggie is going to represent our cell. The inside will be the inside of the cell, and then the baggie itself will be the cell membrane. And then I've got some starch solution here as well, and I'm going to use my graduated cylinder to measure some things out. In terms of safety, I do have some chemicals that I'm using, so again I'm going to use my goggles. And I also have my safety ring at the top of my graduated cylinder. Remember, that's to stop it from breaking in case it does fall over. And so I just kind of be aware of those couple safety issues. In terms of the hypothesis, that'll be on the lab sheet itself, but you're going to guess what you think is going to turn purple. There's going to be a liquid inside the baggie, there'll be a liquid inside the cup that'll be outside the baggie, or you could put both, or you could put neither. So make sure you answer that before you actually go through and do the lab. And so let me just go ahead and get started. Again, this is going to represent our cell, and we're trying to figure out if diffusion actually takes place of movement of molecules going from an area of high concentration to an area of low concentration until equilibrium is reached. And so to start off with that, we're going to just take some starch solution, which I have here. It's going to be 40 mLs, and again I'm going to keep this flat on the table and kind of get down to where I can see it a little bit better. Once I get close, so let me get down to eye level with this. Make sure I get my 40 mLs in there. And that looks pretty good. Okay, so 40 mLs of starch solution is going to go into my bag. And then I'm going to go ahead and seal that. Just double check and make sure everything's good and sealed. And that's going to represent what's inside my cell, and then my cup is going to represent what's outside my cell. So I'm going to put that bag off to the side for just a second. And I'm going to fill this cup about halfway full with water. I don't really have to measure it, just kind of filling it about halfway full with water. And right about there is pretty good. And then I'm going to take my iodine. This is concentrated iodine, so you want to be real careful with this, not to get it on your skin, because it will stain or your clothes, it'll ruin your clothes. So you want to be real careful with this and just make sure that nobody's kind of messing around, stuff like that. And I'm just going to take kind of a dropper full of that, and I'm going to add it to my water. And then kind of stir it a little bit, and I'm going to add a little bit more because that's a little bit on the light side. But I want it to be that kind of like, that dark amber color, kind of dark apple juice looking color. And then I'm going to make sure my cap is back on just to make sure nothing spills. But I want it to be that sort of nice golden, real dark apple juice looking color. That's what we call amber. And so if I look at my before pictures, this is what I'm going to put on my data table. The color of the starch, or what's inside the bag before, I'm just going to call that white because it's kind of cloudy and white looking. And then in terms of the cup, I'm going to call that amber. And then I'm going to let you fill in the after. But I am going to show you the results. Normally what we would do is we would take this and we would stick this in the cup and we would let it sit there overnight, and you would have your group number and your period number on there, and we would just let it sit overnight to make sure you got the same exact cup back. I prepared one ahead of time, so this is to kind of show you what it looks like afterwards. And so you can see that the stuff in the bag turned that purplish color we were looking for. And the stuff in the cup is, if we kind of compare the two, is a lot lighter now. Okay, let me kind of get rid of the bag so you can sort of check that out a little bit. But you can sort of see they started off about the same color, and now this one is the one we just started with, and this is the one that was sitting overnight. And so you can sort of see that it's a lot lighter in color. And so I'm going to let you go ahead and put the after in. Again, this is still considered amber, but I might call it like light amber, something like that, but it's definitely not the same color that it was before, and the baggie is more of a purplish color. All right? And so from there, you're going to be asked some questions after you fill in the data table. I think there's 7 questions, and don't forget to write your conclusion in the CER format. I'm still getting some labs turned in without those CER conclusions. But other than that, that's pretty much the results for our lab. And I hope everything makes sense. If not, please let me know and I'll be happy to clarify. Have a good day.